My title is Demographic, Clinical, and Genetic Factors Associated with COVID-19 Disease, Susceptibility and Mortality in Kurdish Population. This is part of my dissertation in 2020 until 2024. Fortunately, this paper published in Annals of Saudi Medicine. My topics include introduction and materials, methods, results, conclusions, and recommendation. We're starting with introduction. As we know, by the 2nd March of 2023, as declared by WHO, there are more than 61 million people, cases confirmed by COVID-19, and there are more than 6 million deaths around the world, and about more than 13 billion vaccine doses were given until March 2023. And the disease, we have a classification, different classification of disease. According to the Q Young in 2021, we have 5 groups of disease. One of them is asymptomatic or pre-symptomatic. There is no symptoms, but positive by real-time PCR. And we have mild illness. There are some mild symptoms like fever, cough. And we have moderate illness, where we can show by radiographic CT scan of chest. And but still in these 3 stages, the oxygen saturation is more than 94. But in the severe illness group, the oxygen saturation is below 94. And in the later stages, last stage is critical illness, which causes respiratory failure, multiple organ failure, and sometimes most of the disease in critical stage die by the end of the disease. And we have this graph or this diagram created by Byrender by myself, show that the binding of SARS-CoV-2 to its receptor angiotensin converting 2 and the co-receptor TMPRSS2, which is a protease receptor, which is help in binding in SARS-CoV-2 to entering the cell. Inside the cell, we have different receptors like toll-like receptors and activation of various transcription factors. Then there is a release of or transcription large amounts of inflammatory cytokine, among them tumor necrosis factor alpha, and also the interferon gamma has dual, like double-edged swords, has dual function. Previously, interferon gamma has a role to interfere with viral replication, antiviral response to the surrounding cell. But in severe cases of the COVID, interferon gamma has a bad role in the cytokine storm by the activation of T helper or by polarization of naive T helper to T helper 1. Finally causes the activation of B lymphocyte antibody response. And this causes acute respiratory distress syndromes, and also the activation of neutrophil and macrophage releases oxidative stress also has a role in the endothelial dysfunction, tissue damage, and ARDS. So why we focus on the single nucleotide polymorphism of 4 genes in this paper? Because you know the angiotensin converting enzyme is the main receptor for SARS-CoV-2. The TMPRSS2 is a protease, acts as a co-receptors, and intertumor necrosis factor has a role in cytokine storm and ARDS. So we focus on tumor necrosis factor. And also interferon gamma, we focus on interferon gamma because has still negotiating or controversial role in SARS-CoV-2. So as in this study, as we mentioned, we focused on tumor necrosis factor, alpha genes, and we focused on 3 polymorphisms located in the promoter region, and we focused on 4 polymorphisms in angiotensin converting enzyme 2, which are these RSs. And in the interferon gamma, we focused only on 1 polymorphism, which is 24-35-561, which is located in intron 1. Lastly, In the TMPRSS2, we focused on two polymorphisms located in intron 11. Why we focused on this? Because previously I reviewed in many articles, and before of this article I published the review articles about the role of inflammation in SARS-CoV-2. So I have selected these 4 genes and those polymorphisms. Our aim, as we mentioned, we have many predictors of disease and mortality, and these are critical. Why? Because if we, in the early stage of disease, for example, in the entry of the hospital, if we know this patient suffering or has a polymorphism, we must pay attention more than the other disease because it may cause death. So there are many predictors. For example, some of them are clinically like olfactory dysfunction and gustatory dysfunction. This is called chemosensory dysfunction because some patients have no or lose their smell or tastes. And inflammatory markers like CRP, and also we focused on hematological biomarkers like neutrophil, lymphocyte, and the ratio of neutrophil to lymphocyte, which is a new markers of inflammation. And also coagulation markers is another marker. And also immunogenetic markers like these 4 genes and their cytokines, especially tumor necrosis factor, alpha and interferon gamma, because these 2 immunogenetics, we focused on the gene and also we focused on the serum protein. So the aim objectives of this study to examine the role of cytokine, tumor necrosis factor, and interferon gamma in predicting COVID-19 mortality. And also we focused on demographic factors like age, body mass index, and comorbidity with severity of disease. And also assessing routine light biomarkers like CRP, D-dimer, lymphocyte, neutrophil, and NLR linked to the severity and mortality of disease. And also selecting many polymorphism in these 4 genes in survival rate by Kaplan-Meier and susceptibility of the disease. And also, is there any connection between this polymorphism, 2 polymorphism, and the amount of the producing cytokine? So our material method or design of study includes like this picture. We have COVID-19 patients, 953, and healthy control is 50. Why the number of the control is low compared to the healthy COVID-19? Because it is difficult to, because this paper done in 2021, it's difficult to find the control in that period. So the COVID-19 admission to the hospital, all of them, and we have divided into subdivided in 2 groups: survived case, 105 cases, and non-survived includes 48. After a collection of data and sample and the COVID-19 patient confirmed in the hospital by real-time PCR, we measure laboratory markers, CRP, D-dimer, TNF, interferon gamma, and lymphocyte, neutrophil, NLR, TNF and interferon gamma by ELISA, while the others by COBAS, lymphocyte, neutrophil by Coulter, and NLR by dividing lymphocyte to neutrophil ratio. And the data like demographic factors, for example, age, body mass index, olfactory dysfunction, or gustatory dysfunction. And also we do SNPs of the genes. I have designed primers by myself for these 4 genes. And also we sent these samples after PCR products to the Korea to do Sanger sequencing. Then after that, we do survival analysis, Kaplan-Meier and Cox's regression to know the predicting of severity and susceptibility and mortality of the disease. This is the design of study. This is another diagram of study. We took the blood and divided into serum and the whole blood. That's from the serum CRP and from the plasma D-dimer, and also we measured TNF and interferon gamma by ELISA. And the whole blood we do by Coulter neutrophil lymphocyte and NLR by calculation of neutrophil to lymphocyte. And from whole blood DNA extraction, then nano drop to know the quantity and the quality of DNA, then do PCR and gel electrophoresis, then do Sanger sequences in Korea. So, and this is the gel electrophoresis results for angiotensin converting enzymes. The band 456 pair, it means the gene of present. And also this is the TMPRSS2 gene, which is 584 base pairs. And this is tumor necrosis factor. And lastly, we have interferon gamma. Okay, statistical analysis. We do normality tests. Our tests do not obey the normality tests, so do non-parametric tests. And we do ROC curve. And also we do long rank test and Kaplan-Meier method. The hazard ratio, it's like odd ratio to what's found in the Cox regression to know it's a risk factor or not. So the results in these tables, we divided the group of participants into healthy controls and into COVID-19 to know this factor has a role in susceptibility of disease or not. We see that the red one is significant. For example, the gender male acts as a risk factors. Male has more susceptibility to the COVID-19 due to habiting and due to smoking, more alcohol, and more comorbidity. And also the D-dimer is more than in the male than the female, and TNF, in the COVID-19 compared to the control, and also interferon gamma also, but it's less than in COVID-19 compared to control. This is maybe due that this is a good role interferon gamma, so it's less in COVID-19, but in some other cases, the interferon gamma increases. And also neutrophil and lymphocyte and NLR also increases more in, lymphocyte decreases while neutrophil increase. Why lymphocyte decreases in COVID patients? Because it's called lymphocyte exhaust. Due to the combat or the fight between the immune cells and the corona, the SARS-CoV-2 causes the exhaustion of the lymphocyte. The receptors cannot activate it very well, so cannot present the SARS-CoV-2 to other cells, so causes decrease the immunity. And the other classification of disease between survived and non-survived, we see that the more ages causes more non-survived. Why? Because due to presence of comorbidity, and we see that the D-dimers also more in dead patients compared to survived, and also all other parameters, CRP and interferon gamma, NLR, more of them increases except lymphocyte will be increased in dead patients due to lymphocyte exhaust. And uses Kaplan survival analysis, especially Cox regression, to find hazard ratio and regression coefficients. So we divided the models into less than and equal to 40 and more than 40. We see that more than 40 is a hazard ratio, which is 2.5. It means the patients which are more than 40 is 2.5 more probability to increase his death compared to less than 40. And we see that the comorbidity, the patients with other diseases has 2.6 is more than susceptible to death than patient that has no comorbidity. For that reason, we don't exclude the comorbidity or presence of other disease besides COVID-19. The others are non-significant. And this is the ROC curve for knowing the mortality. We divided group into two groups, survived and non-survived. The red one, it means this is high predictors for severity or mortality. Why? Because if area under curve and the ROC curve is near to the one, it means has more predictors or strong predictors of mortality. NLR, tumor necrosis factor, and interferon gamma is more predictors of the disease. About the roles of 4 genes and polymorphisms of these 4 genes in susceptibility of disease, we divide groups into healthy controls and COVID-19 by doing chi-square tests. We found we showed in this table only significant results because angiotensin converting enzymes and interferon gamma has no roles in susceptibility of disease. Only TMPRSS2 in RS220, 70, and 788 has a roles. As means we mentioned that, for example, this is odd ratio. The GA is more less than 1, and AA is more is 0.2. So if the odd ratio is less than 1, it means it's protective factor. So this gene, this polymorphism, it presents increased resistance to susceptible to the COVID-19. Okay, less susceptible to get COVID-19. Whilst the tumor necrosis factor alpha in this polymorphism, 180.0629, we see that the odd ratio is more than 1. It means risk factor by chi-squared. So GA and A is a risk factors. For example, the patients had GA 3.4 is more susceptible to COVID-19 than the GG genotype. This is about the susceptibility. But about the mortality, predicting mortality, we have used univariate Cox regression, and we concluded that the combination, the GI genotype in the polymorphism of tumor necrosis factor 180.8629 has hazard ratio significantly 3.8. It means the patients with this genotype are more prone to death, 3.8 more than compared to the GG genotype. And also interferon gamma, also we see that the TT genotype, as we see in this Kaplan-Meier survival, we see that, for example, the violet colors is a GG genotype of tumor necrosis factor, while GA is a green one, is intermittent line, is a GA genotype. We see that the survival, Kaplan-Meier show that the survival line decreases. It means that the probability of survivability of the GI genotype is less than the GG genotype. And also, if we see the interferon gamma in this polymorphism 3561, the TT genotype also reduces the survival of the COVID patients. So the patient has GI genotype or TNF and TT genotype of interferon gamma of this polymorphism reduce the survival. It means increased mortality of disease. In order to know if there is any association between the genotype of TNF and increasing this corresponding cytokine, we have shown that we see the GI genotype significantly increase TNF and also TT genotype increase the cytokine interferon gamma. In conclusion, we see that the first one is not true. The second point is true. Age and body mass index and comorbidity risk factor for death in COVID-19, and routine laboratory marker like CRP and D-dimer associated with COVID severity by ICP hospitalization and CPO2 dysplasia, and the TMPRSS2 and TNF alpha, those SNP are altered susceptibility to SARS-CoV-2. TMPRS2 decrease susceptibility, while TNF increase susceptibility to COVID infection, while these two polymorphisms associated with increased mortality and decreased survival rate. And also these two polymorphisms also increases the corresponding cytokine. By this mechanism may contribute to the severity of the disease. In the recommendation and the future works, last slide, it's better, for example, some polymorphisms in ACE2 or in TMPRSS or tumor necrosis factor are close to each other. So it's better to do linkage or haplotype, but unfortunately, we did not do this. In the future, inshallah, if there is another pandemic, we can do further research on it. Thank you very much.